Hello everyone, let's begin with another topic Introduction to Nucleic Acids. Nucleic acids are essential biomolecules that carry genetic information. They are responsible for storing, transmitting, and expressing hereditary traits in all living organisms. Let's begin with structure of nucleic acids. In structure, we have nucleotides, nitrogen bases, polynucleotides, and first one, nucleotides. It means the nucleic acids are composed of monomers called nucleotides. Each nucleotide consists of a sugar molecule, a phosphate group, and a nitrogenous base. Nitrogenous base means in this we have five main nitrogenous bases: adenine, guanine, cytosine, thymine, and the uracil. These bases pair up in a specific manner, forming the basic of genetic information. Polynucleotide chains. These are linked together by phosphodiester bonds, forming long polynucleotide chains. These chains can be either single-stranded or double-stranded. In First, let's begin with deoxyribonucleic acid, which we generally called as DNA. Genetic blueprint. DNA is the primary carrier of genetic information in most organisms. It contains the instructions for building and maintaining an organism. Double helix structure. DNA is a double-stranded helix with two polynucleotide chains running in opposite directions and held together by hydrogen bonds between complementary base pairs. Base pairing: adenine pairs with thymine and guanine pair with cytosine. This complementary base pairing is crucial for DNA replication and transcription. Chromosomes: in eukaryotic cells, DNA is packaged into chromosomes, which are tightly coiled structures that ensure proper segregation during cell division. Ribonucleic acid, messenger RNA, which means mRNA, it carries genetic information from DNA to ribosomes, where proteins are synthesized. It is a single-stranded molecule that serves as a template for protein synthesis. Transfer RNA, which is known as tRNA, it is the molecules transport amino acids to ribosomes during protein synthesis. They have a specific anticodon that recognizes a codon in mRNA, ensuring the correct amino acids is incorporated into the growing polypeptide chain. Ribosomal RNA. rRNA is a structural component of ribosomes, the cellular machinery responsible for protein synthesis. It plays a crucial role in the assembly of ribosomes and the translation of mRNA into proteins. Genetic information storage and transfer. DNA replication. Before a cell divides, its DNA must be copied. This process is called replication, and it ensures that each daughter cell receives a complete set of genetic information. Transcription. DNA contains the blueprint for protein synthesis, but it is not directly used to make proteins. Instead, the genetic information is first transcribed into mRNA. Translation: mRNA carries the genetic code to ribosomes where it is translated into a protein. This process involves the interaction of mRNA, tRNA, and rRNA, resulting in the assembly of polypeptide chain. Protein synthesis. Initiation. The process begins with the binding of mRNA to a ribosome. tRNA carrying the first amino acid binds to the start codon in mRNA. Elongation. The ribosomes moves along the mRNA reading the codons one by one. Each codon is recognized by a specific tRNA carrying the corresponding amino acids. Termination. The ribosome reaches a stop codon in mRNA, signaling the end of translation. The polypeptide chain is released from the ribosome and folds into the functional protein. Replication and cell division. DNA unwinding, primary synthesis, DNA polymerase action, ligase action. These are the some of the replication. In this, the cell division happens through the double helix unwinds and separates into two strands. In primary synthesis, short RNA primers are synthesized, providing a starting point for DNA polymerase. In DNA polymerase action, DNA polymerase adds nucleotides to the new strand using the existing strand as a template following the base pairing rules. In ligase action, DNA ligase joins the newly synthesized fragments of DNA, forming a complete double-stranded DNA molecules. Transcription and gene expression. DNA unwinding. RNA polymerase binds the DNA molecule at specific site called the promoter. RNA synthesis. RNA polymerase moves along the DNA template strand, synthesizing a complementary RNA molecule using the base pairing rules. Termination. RNA polymerase reaches a termination signal in the DNA sequence, releasing the newly synthesized mRNA molecules. Regulations of cellular processes. Transcriptional regulations. Transcriptional transcription factors bind to DNA sequences, either activating or inhibiting the transcription of specific genes, controlling the expression of genes. Post-transcription regulation. It means RNA processing includes splicing, capping, and polyadenylation affects the stability and translation of mRNA molecules, influencing protein synthesis. Translation regulation. MicroRNAs can bind to mRNA molecules, inhibiting their translation, playing a critical role in regulating gene expression. Post-translation regulation. Protein modifications such as phosphorylation, acetylation, and ubiquitination can affect to protein activity, stability, and localization, influencing cellular processes. So lastly, importance of nucleic acids in biological systems. Genetic inheritance. Nucleic acids carry and transmit genetic information from generation to generation, ensuring the continuity of life. Protein synthesis. Nucleic acids provide the instruction for building and maintaining all the proteins necessary for cellular functions. Cellular regulation. Nucleic acids play a crucial role in regulating cellular processes, ensuring proper cell division, growth, and development. Evolution. Changes in DNA sequences over time, known as mutations, are a driving force behind evolution and the diversity of living Earth.